Good morning, good afternoon, or good evening, depends on when you are on Earth. So I'm going to talk about something that we have, well, an initiative we have recently started, of course, like most of you, I think, here, which is COVID on the web. Basically, that started as some initiative where we wanted to experiment some tools that we had on some COVID-related data. And soon enough, that became something bigger, and we felt like, okay, that's actually a real project starting. So just a word before about the team. So I'm from a team called WMIX, Web Instrumented Man-Machine Interactions, Communities, and Semantics. So it's a big, long name, but the motto of the team is that one that you have. So
Frank, I'm sorry, this is David. I have to interrupt for just a silly administrative thing, but I just remembered we are recording this, so I need to inform everybody about that. Yep. So we are recording this, everybody. So if anybody does not want to be recorded, then either keep yourself muted or drop off. So I'm so sorry that I forgot to say that earlier. Okay, go ahead.
No problem. Okay, so yeah, the motto of the team is this one: bridge formal semantics and social semantics of the web. So formal semantics, understand knowledge engineering and reasoning, the whole semantic web stuff, basically. And social semantics, obviously, is going to be much more on the study of communities on the web, social produced data that is mostly text, but not only. So the idea behind this project was, okay, among all the things that we can do in that team, okay, what is it that we could do if we apply those tools to the COVID-19 related data? So so pretty soon we found out the CORD-19 dataset, and so the main goal here is to, yeah, like I said, adapt and combine the tools that we have, that we're used to working with, and take that to make it easier for biologists or maybe medical people access and make sense and analyze and make sense of COVID-19 related data and related literature in that case. So we have identified several leads. Say the first one was about identifying named entities from the text of the articles, that is the 47 or so thousands articles which are in the CORD-19 corpus, and see what we can do with that and how we can allow people search the corpus using named entities. The second axis was about argumentative graphs, that is the ability to extract automatically arguments from text, detect which component, which say argument is claimed, what argument supports or attacks another argument, and so on, and see what we can do with that. These two axes, there is some orthogonal approach to that, is to provide visualization and exploration tools that are relevant, suitable for that kind of data. And the last thing is that our background is about semantic web and linked data. So we want to make all of this published as linked open data and accessible to the whole community. Okay, so let me start with the first part about identifying and disambiguating named entities. So basically, we have started with state-of-the-art tools, which are, you probably know, DBpedia Spotlight, anti-phishing maybe, that does more or less the same thing as DBpedia, but it disambiguates named entities against Wikidata and Wikipedia at the same time. So it uses Wikipedia articles as a context to make sure the URIs it identifies for Wikidata or to improve, not to make sure, but to improve the confidence of the IRIs it identifies from Wikidata to relate to named entities. And the third one is the NCBO bioportal annotator, which makes this same disambiguation against all the wealth of ontologies that are in the bioportal. So I'm going straight to the results, and I will give you a bit more details afterwards about how it is provided. But basically, we are in the process of releasing a second version, 1.1, of our dataset. And altogether, there are 54 million named entities that have been identified by the 3 tools. And that amounts to over 500,000 unique URIs, either from DBpedia, from Wikidata, or from bioportal ontologies. So the highest number is with Wikidata, because with DBpedia and bioportal, so far we have been able to process only articles, titles, and abstracts. We have been able to process the body with Wikidata, but that's quite massive, and doing the same thing with DBpedia and with, say, the result of Spotlight and the annotator of bioportal is really huge. So that's something we maybe will discuss in I don't know, in the discussions, but we are dealing with you know, massive amounts of data, and yeah, providing hardware that can and software that can support querying all those entities and make sense and reason on all of that, it's pretty challenging. So just a few details about the dataset itself. So we call that the CORD-19 named entity knowledge graph. So we have 2 parts: articles metadata, common metadata, which was about the title, the authors, DOIs, other identifiers, and so on. So for that we used, you know, classical ontologies, Dublin-Core, Fabio, Bibo, so on. The more interesting part here is about named entities, and you can think of named entities as annotations. That is, you consider text, which is an article, and you say at that spot, precisely in that text, we identified that this word or these 2 words are a named entity. So you can annotate the article and say that specific piece of text is related to a URI in DBpedia, Wikipedia, and so on. And that's exactly the point of the web annotations ontology that allows you to annotate any kind of document of the web, whether this is text or image or recordings or whatever. So here, this is a quick example of how the RDF looks like. So this blank note here is an instance of annotation. We related to the article. This is here. Can you see my mouse, by the way? I'm not sure if I'm moving here, if you can see that.
I can.
Yes. Yes.
Okay, good. So the schema about just basically relates to the URI of the article. And then, so we have the confidence of the named entity disambiguation. The body is the URI. So here it's the Wikidata URI. And here is the piece of text that has been identified. So it's the has target priority in the web annotations ontology. The source is, oops, I lost my mouse. The source here says that this was found in the abstract of the article. So we have not only an IRI for the article, but also an IRI for the title, the abstract, and the body. And then this part says that within the abstract, we have found this text PCR at offset 235 to 238. By the way, PCR is a polymerase chain reaction for those of you who are not biologists like me. And basically, this is exactly the URI of that stuff here in Wikidata. So basically, this is it. The model is not very complicated. We just provide one such instance of annotation for each of the named entities that have been discovered by Spotlight, entity fishing, and the annotator of bioportal. But that's pretty big. So you have given a few details here. You have a full detail about the RDF modeling. There is a Sparkle endpoint that you could use. You can also use the Virtuoso faceted browser here, and we have put the tools and everything on the GitHub. Okay, but now what? Okay, we have named entities. What do we want to do with that? So typically, this is the kind of question that biologists and doctors would like to ask. That is, what are the articles from the corpus, the CORD-19 campus, that would mention at the same time a cancer and a virus of the coronavirus family? So that means not only the URI of cancer, but any kind of cancer that would be probably classified under some class of cancer or something like that. And this is the kind of thing we have tried to go so far to do. So here is an example. Here, this is the entity, the cancer entity within Wikipedia, Wikidata, sorry. And this is the entity for the coronavirus family of viruses. So what we've done is that we have run a query that reasons on the definition of that class here of cancer and that class of coronavirus, and that tries to find an article that would have annotations for any of this one or its subclasses or instances of its subclasses. And basically it gives quite a lot interesting results. So this is just a subset here, but you see this is the article URI, the title, and here you can see what kind of cancer has been annotated and what kind of virus has been annotated. So this is the kind of thing we are still working on. That is, we are exploring with people from the INSERM in France, which are the the equivalent of the NIH. So what are the questions that they want to ask, and how they want to exploit this kind of thing to explore the data set, the CORD-19 data set, and find, I don't know, evidence that would help them in finding out curation or things like that. Okay, second thing, wait a sec, I think I have a problem in the order. Okay, this one is probably to forget, I think. So I also mentioned something about visualization and exploration of the data set. So we are working with some people who are specialized in graphical interface and more generally user interactions. So this is an example of a library, a visualization library that they have developed and that we are using here. So basically the query I just mentioned before, what are the papers that mention both a virus of the coronavirus family and a cancer? This is a visualization of the result. Here you see the red spots are instances of a virus of the coronavirus family, and blue spots are instances of a cancer. So basically you have these points, these dots show papers, articles that mention one or the other or both of the concepts, that is a virus and the cancer. And then you can basically right click on each node, and then you will get to different types of visualization, that this one on top and this one on the bottom. This one will give you co-occurrences. So here I have clicked on one of the nodes, which is severe acute something. I don't have the full title here. And it gives me the co-occurrences of this term with other, so in this case, this is a respiratory disease. And I will have co-occurrences with the other term, other kind of concept I have asked here. So that allows you to find which papers say mention that disease together with one of these other disease. So it's basically a visualization type that has been designed initially to see networks of co-authorship. So it's something we are still in the process of adapting, but it's pretty powerful. So you have this co-occurrence of concepts in the papers, and here the length of that bar gives you the number of co-occurrences. That is, the bigger it is, the bigger the number of papers that mention both this concept and this one at the same time. And here you will have some other visualization that will help you visualize more clusters. That is, not only I will know that this one is co-mentioned with this one, but maybe it's also co-mentioned with this one. So this one here will give me this kind of visualization, and I can, like that, continue exploring. Each time I can right-click on the node and go further and so on. So yeah, I'm not saying more about this. This is really ongoing exploration work, but we're pretty confident that it has really good. There are really interesting things to do here. And the second thing I wanted to mention here, so I'm pasting slides from colleagues of mine. This is not my part, so honestly, I will be less, say, verbose on that one. So ACTA stands for Tool for Argumentative Clinical Trial Analysis. So basically, it's an approach that's done by my colleagues here, Tobias Meyer, Elena Cabrillo, and Serenari Villata. The point is to automatically extract arguments from a text, arguments that is claims or evidence, and be able to relate those arguments. That is, be able to say that one claim is supported by an evidence, or on the contrary, it is attacked by another claim or by another evidence. So there are intra-relation arguments and inter-paper relationships. So there is a website. Basically, it's been developed really to work on clinical trial corpus, that is texts, that is scientific articles that report the results of clinical trials. And we are in the process of seeing how we can adapt this. So should we focus on the clinical trial papers within the whole CORD-19 corpus, or can we widen the analysis? Do we bear the risk of providing many false positives? This is not so clear yet, but analyzing a much wider type of articles is definitely not easy. So the adaptation is not straight away, but there are some interesting leads here. And basically with that tool, what you could do is to select papers from PubMed, for instance, and then run an analysis of the arguments, and then display, well, that is visualize the set of arguments, and it will look like that. So here you have a graph of arguments, claims, and evidences. Each claim is here on the left. You have an identifier of the paper where it was identified, and on the right you're going to have an extract of the text with highlighted in blue for the claims and orange for the evidence, and then you will be able to browse like that, move from the claim to an evidence that supports that claim, and so on. So this is the work we're in the process of doing. Again, this is exploratory; it's not done yet so far, but we have pretty good hope that we can really get interesting material from this analysis. Okay, so if I wrap up a little bit, so we've started from the CORD-19 corpus with two leads. First is the named entities extraction, and the second is the argumentative graph extraction. Our point is, so the first thing is to enrich the whole CORD-19 dataset with this information extracted, be able to perform some different types of visualization and exploration, and say behind the curtain, do some reasoning so that we can have clever way of answering questions. In the future, we want to continue this work and try to integrate this further with other data sets, not only CORD-19, but maybe other data sets, and see what interesting use cases we can find out. So this is very much driven by the discussions also we have with other people from the more healthcare institutes. And a main constant concern we have here is to do that as to publish the result of all of this as linked up in data. So basically, if you look at this quick list, we are addressing more or less all the letters in the FAIR acronym. So saying you're doing some FAIR thing is maybe, I don't know, a bit of cliche now. But still, it's very important. So specifically in that situation today with the pandemic, focusing on these FAIR aspects of the way you provide data and you make it queryable and iterable and reusable, this is of probably the utmost importance right now. And I think this is it. Yeah.
Wonderful. Thank you, Frank. So we have plenty of time for questions. And let's see, I think, just trying to see if there's a question mechanism or, no, I think if you want to, if you have a question, I think you can just unmute yourself and speak up. I'll start off with a question that I have, which is, what ontologies are you using for determining subclass relationships of cancer, for example? That was an example that you used earlier.
So for now, we've done, for now we have done that only with Wikidata, and basically we have used two things: the standard subclass of property in Wikidata. I don't know by heart its ID, but there is a subclass and an instance of must be P31, and the other one is maybe 273 or 279 or something. Anyway, so we are just collecting all the URIs that have been identified by entity fishing, so the Wikidata URIs, and then we are querying Wikidata to get all the hierarchy of classes, parent classes and subclasses for that URI, so that we have we sort of extract a subset of Wikidata focused on the URIs that have been disambiguated, and then we can reason of that on top of that. Just there is one exception to that, which is very important in that case. The viruses, whatever they are, in Wikidata are not modeled as classes but are taxa, and there is a taxa class and probably which might be parent taxa. So what we've done is basically to get that and regenerate this as a subclass of relationship so that we can reason on viruses as well as cancer or any kind of pathology the same way.
Okay, so you're not using anything like SNOMED or anything like that?
No, not so far, no.
Okay. Okay, yeah.
Yeah, hi, Victor here.
It's Martin. Go ahead.
Okay, thanks. I'll be quick. Yeah, so I have two quick questions. The one is, could you specify a bit why you're not using full text for DBpedia Spotlight, or the other way around? Why can't DBpedia Spotlight process full text? And the second one is, these offsets that you showed in your piece of RDF, are you somehow publishing the, let's say, the canonical text? So if I find these offsets, how can I be sure that I'm aligned in my own version of the dataset?
Okay. So first question about the problem of processing the whole text. So, like I said at the beginning, we are at the moment we have 54 million named entities identified, and altogether we have a graph of 700 million triples. I don't know for you, but for now I don't have the machine to deal with that. We are not Wikipedia or Wikidata, so we have to move to a bigger machine. Basically, this is on the way, but so far it's really too big to process and upload that in our endpoint. But that's only purely a question of machine sizing. There's no other limitation than that. Your second question was, sorry. Oh yeah, what it refers to when I say it's offset 235 in the abstract and so on. So it refers specifically to what's provided by the CORD-19 dataset that is the result of the Gobid analysis. So the CORD-19 contains the title, abstract, and body of the articles. And this is exactly what we have given to Spotlight or entity fishing or annotator. So the offsets are specifically against those fields as they were processed by Gobid and provided by CORD-19.
And in the case when there, for example, the body consists of several paragraphs. I think the 19 dataset comes spread with paragraphs.
Yes, that's correct. In that case, we have just stuck together one after the one, all the pieces of the body. But
Okay.
yeah, you're right. That should be described somewhere.
Okay, thanks.
Hi, it's Martin from LVL. So I guess I have two questions. So the first one is, how are you extracting information about the viral proteins? So, you know, the individual protein products that get produced from the polyproteins.
So, sorry, I didn't get that. How I extract which information?
The information about the proteins in SARS-CoV-2, right? So the viral proteins, because, you know, there's the polyproteins that gets cleaved into the protein products, right? And then those in the end are the interesting drug targets, like the main protease or the RNA-dependent RNA polymerase, right? Do you have any thoughts on that?
So I'm definitely not a biologist, so I'm not sure I understand what you mean here, but technically, technically, yeah. We fully rely on the results of the three tools that we're using. So if some proteins are identified in disambiguating correctly against Wikidata or DPpedia or Semantic in CBO, we'll have them. But I'm afraid I cannot say more than that because I don't know anything about how proteins work.
I see. But say, you know, say an article mentioned, you know, a gene name or protein name NSP1, right? Would that be an entity that would be extracted and represented in your data, do you think?
I do think so. Yeah, very likely, honestly. I could double check with some colleagues of mine who are much more skilled on this kind of issue, but you can also give it a try. I mean, make a few queries and see what you get.
Yeah, I work with the gene ontology people at the Lawrence Berkeley Lab, and I guess what
sorry,
how you get information on the actual proteins in this virus.
Marcin, you cut out for a moment. Can you say that? Can you say your question again? You cut out for a moment.
Well, I guess it's just a comment that I work with the gene ontology group at LBL, and one of the challenges has been to link, you know, all the information to the specific proteins in this new virus. So that's kind of ongoing effort. I think it's a good challenge for everyone to think about. The other quick question I had was, you know, there's a few of these different entity extraction and NLP efforts for COVID, and I guess I'm wondering now that there seems to be enough of them that maybe we need some kind of overarching, you know, community at least, or maybe even an effort to merge these different annotations, right? Do you have any thoughts on that?
Yeah, absolutely. Actually, if you look at last week's presentation from Victor, it was pretty much the same goal, that is annotating a corpus and reason on that. And the week before we had, I don't remember his name, but there was a much more long-lasting project about annotating data sets in general. And so indeed, I think named entities recognition and annotation is a pretty common need. So we are several groups to do kind of the same thing with maybe somehow different tools. But at some point, yeah, there will be a need for a way to, I don't know, not organize, but maybe something less strong that word, but federate the efforts towards the same goal. Although I don't have any leads as to how to do it for now, honestly.
Yes, this is David. Yeah, Marcin, that's exactly the motivation behind these teleconferences, is to start that process of just getting people to know about each other's work, and then we'll go from there to figure out, you know, how do we best coordinate the efforts.
Perfect, thank you. Do you know if those previous presentations are available somewhere still from the last week?
Unfortunately, the other ones are not. We only just started recording them, and I apologize to do that previously.
But now the slides will be available, I guess.
Yeah, we do have slides available, and we have the notes available too in the notes document. The notes document contains all the notes. Yeah.
Yeah, sorry, I just posted. Could you actually post the link to the notes? I had a crash earlier.
Yeah, sure. I'll put it in the chat again. Yep.
All right. Thank you very much.
This is Michael Liebman. I have a question. In our experience, we noted two things, because you're talking about sometimes using the body and the abstract, and sometimes the abstract. We did some studies where we compared analysis of abstracts against analysis of full body articles. And we were doing that frequently to potentially pull out ontologies at the time, so sort of semantic mapping. And we found a significant difference in general because the abstracts, of course, they tend to be more frequently of an advertisement for the article than a real summary of what's in the article. And so the confidence of the extraction from that we found to be different from the confidence from the full body. And then the other observation that we had was that in dealing with the full body, we also found it necessary to parse an article so that we separated the section on methods, the section on results, and the section on discussion, because obviously what's put in discussion is conjecture and not necessarily proven facts. And we found that, again, the differences could be significant, especially when you're trying to extract knowledge from it, not just graph the terminology that's being used.
Yeah, so yeah, I definitely understand that your comment. And but by the way, if you can give us the reference to that study, I'd be very interested in that. Maybe just
Michael, if you could stick it into the notes document, that would be great.
Yeah. Just maybe a complementary comment. So talking with my colleagues who are working on our argumentative extraction, graph extraction, the quality, say the representative quality of an abstract seems to be varying a lot from one category of article to another. What they have noticed so far, but it's I'm not sure it is backed by some clear proof or evidence, it's just maybe empirical, is that clinical trials usually have an abstract which gives pretty much a few clear statements about the results, and it's relatively easy to extract some arguments with confidence from the abstract. In some other articles that may not be the case, and maybe like you say, it's going to be more an advertisement of the article, and yeah, that's an issue. And honestly, here I don't know. Well, I'm not involved enough in this kind of research to give leads about how to go there. But for sure, as of now, we have kind of used brute force method. That is, we have taken these 3 annotation tools, run them, process all the articles, and this is it. But probably there was a lot of things to do. Maybe filtering before, or that is like in NCBO, you would like to probably select the ontologies of interest before instead of doing it afterwards, because afterwards, just in the case of this corpus, we have 20 million annotations by NCBO just on the titles and abstracts, not even talking about the body. So that's already huge. So probably filtering is necessary, and segmenting the body, like you suggest, as a hypothesis and methods and results and so on, would probably be necessary. But yeah, there are a lot of things probably to do here.
Other questions, people?
Yeah,
I do have a question. Oh yeah, go ahead. Who's that?
Sorry, it's Victor again. Just a quick comment regarding this federation, or that was, or let's say, avoiding this duplication of efforts that was mentioned earlier. So in our experience, and I guess I'm not the only one saying this, so there is quite some effort in doing these annotations, setting up the pipelines and so on. Some are better prepared than others. Some have better infrastructure than others, but even for the most prepared, it takes a couple of days. It took a couple of days or weeks to go from the release of CORD-19 to the publication of a knowledge graph. So I think one has to consider this fact so as to because I like these kind of meetings where we present, let's say, results. But by the time I have by the time Franck has the nice results he's shown us, a lot of us have already invested a couple of weeks, some of which have been duplicating his work. And the same goes for all of us. So I think it might be also a good idea to kind of, I don't know what to suggest, but to kind of inform each other of the efforts we're going to take before we spend a couple of weeks annotating, so as to better distribute the work. So, I mean, in the case of Franck, for example, the really nice stuff with DBpedia and Wikidata, as far as I have seen, no one else has done it. But the stuff that we have done using gene ontology, for example, it took some time to set up, but other people had also done this. And we only, we found out that others had done this when we had already been working for 1 week and so on. So this is, or 2 weeks. So this is kind of what I would like to maybe comment on, this need for more prompt communications.
Yeah, I agree. That's an interesting comment. And just even about just for instance, the interest of taking into account a new version of CORD-19 or not. We are, I think, so far at version 12 of the dataset. The last time we have used it and generated our own dataset was on version 7. And I honestly don't know so far. I have not checked whether it's useful, worth trying to move to the latest version, but there is one version every week, and there is no way we can keep up with that pace. So this kind of feedback could also be interesting to share. That is, what have you found new in that data set? Was it worth, or can we wait until, I don't know, the version in 1 month from now? That's very practical and pragmatical, but.
Yeah, and this is David. Yeah, Victor, I think you asked a great question there. And actually, sorry, was that Victor or who was that?
Yes.
Okay, yeah. And that is, when we first started these calls, what we did was we actually had very short presentations by everybody that was on the call in order to talk about what they were not necessarily already had done, but planning to do. But then once we had done that, a round of that, we then switched over to deeper dives into what each person was doing. So maybe it's time now to jump back again and do another round of those very short presentations of what each person is planning to do. I'm not sure the best way to coordinate this, and so I really want to hear people's ideas and discussion about it. But that's certainly one thing that we could do on these calls is we could do sort of another roundtable of what each person is thinking of doing for, each person speaking 5 minutes or something like that. Any thoughts about that from anybody?
That's a good idea, and I guess we would volunteer. So I think the idea with the KG COVID repo on GitHub is that it's a mix and match pipeline to build knowledge graphs from ingest that are crowdsourced, and so you can control what goes into your knowledge graph. And I guess maybe my question should have been: do you guys already have a place where you can share the results of your work, right? So you're either the knowledge graphs or the annotations or the named entities, or any of those pieces. That we'd be very interested in that.
Yes. So we do have. Well, so the assumption at the moment is that each group is making their work available on their own site, but we do have. I have collected also a short description of each of the projects that I know about, and we stuck it also on a wiki page on our site, just for lack of another place to put it. And let me get the URL for that and paste that into the chat as well. I think it's at the top of the notes document. It should be at the top of the notes document. Yes, it is at the top of the notes document, but I'll paste it into the chat window as well. Mm-hmm. So that has not been updated as much as it should. So that's due for updating. So anybody that is doing work that is not represented there, please, if you can, do, I'm not sure the mechanism you'll have to do with the wiki permissions or whatever. I guess either just see if you can request changes to it. And if you can't, then in the worst case, you can email them to me and I'll stick them in. Yeah, thanks for the link there. Okay, so here's the link in the chat for where we have the short descriptions. So yeah, okay, so I would be happy to schedule, we could schedule next week to do another roundtable of each person speaking 5 minutes on what they're doing or planning to do. Am I hearing sort of, I heard one person speak up saying, yes, that's a good idea. Do I hear other thoughts about that from others?
I mean, I would gladly speak up for 5 minutes, but I think it's too soon after I had a big presentation.
Yeah, it's too, sorry, it's too soon for you to do a big presentation. Is that what you're saying?
No, I mean, I think most of the people that are here today were also here 2 weeks ago. So I don't think I can bring anything new.
Yeah, well, let's see. I am counting, I think, 1, 2, 3, 4, at least 5 or 6 or 7 people that I don't think were here last week. So you know what, I'll tell you what, why don't we, maybe we should ask for the ones who haven't spoken at all to give a very short thing next week. Maybe that would help. So let's see. Let me run down the list and see who would be interested in doing that. I'm just going to call off names. Frank, you just did yours. So Albert, would you be interested in doing like 5 minutes next week?
I don't think I have anything to offer at the moment. I'm offline.
Okay. Yep. All right. Yep, that's fair. Chumming, would you want to do 5 minutes next week?
Not really. I can talk about the simple couple idea, but
okay.
I don't have anything ready yet.
Yeah, okay, all right. Daniel?
I'm not sure of anything worth presenting, but I can present something else.
So I think I heard that as a yes.
Sure.
A meek yes. Okay, Deborah, you want to speak 5 minutes next week? Deborah, I think you're muted, or let's see, I've unmuted you.
Yep, sorry, I was muted. Yeah. I would really like to catch up on what has been talked about. Do you have the notes? Because this is the first time I've joined. This has been on my calendar forever, but this is the first time I've joined. So I don't really know what anybody else is doing. So
okay, yeah, good point.
I would really like to see notes or summaries from that. And we're really just starting, we're building on stuff that we've already done, but we're really just starting our journey. But I can say where we're going.
Okay, good. Yep. So that's a fair point. So maybe we should, maybe everybody that is doing something or planning to do something should have a 5 minutes, even if they did it previously. So let me continue down the list just so that I get a full list of everybody. So Gaurav, do you want to? Can you speak for 5 minutes on what you're doing, or next week? Are you interested in doing that?
Yeah, sure. I won't, as everyone else has said, it won't be very interesting. It'll just kind of, we're just doing very basic entity extraction, but I can tell you what we're doing.
But it's a start. Yeah. Okay, great. And Golem, you want to do another 5 minutes next week?
Yeah, I think I can do. Previously, I just present, but now I am in the middle of the name entity recognition work with concept names. So I think I can present for 5 minutes.
Okay, Louis, do you want to present for 5 minutes next week?
I don't know that we have enough to really talk about yet. I'm more
okay.
Just
that's fair. Yep.
And feel everybody else's knowledge graph at the moment.
Yep, totally fine. Yep. Marcin Joachimek, do you want to?
Sure, yeah, no problem. And actually, I was going to say we could also volunteer John Daglin from LBL to talk about COVID Scholar. Could be interesting for you guys.
Okay, what was the name again?
John Dagdalen.
Okay.
And it's covidscholarly.com. I posted a link in the chat.
Okay. Oh, good. Okay, thank you. Michael, do you want to spend 5 minutes next week?
Sure, that would be fine.
Okay, great. Subhashish, you want to speak for 5 minutes next week?
I can do next, but this time, no.
Okay, all right. So not next week, but the following week.
Yes.
Okay. And Tom, do you want to speak 5 minutes next week?
I certainly could, but at this point, I've done a few little things on COVID specific, but for the most part, right now, I'm just trying to keep the standard Monarch knowledge graph updated, and it seems as though everybody and their mother is updating their data sets, and so I'm pretty much in the weeds, just rebuilding existing resources for people.
Okay. Would it be better to do it at a later week? Or what?
I haven't noticed a change. I mean,
okay. All right.
I'm in a grind. Okay.
All right. So I think I'm taking that as a yes, if I've interpreted what you said.
Sure. I mean, I don't know. I can try to familiarize people with Monarch if they aren't used to it already.
Yeah. Okay. Yep. And I can do 5 minutes on what we're doing at Mayo Clinic and Johns Hopkins as well. Okay. So we've got a plan for next week. We're running out of time now. We're just about at the end of the hour. Are there any comments that anybody wants to make, or any final questions that anybody wants to ask?
I have a request that when people present, maybe they can say what of their work might be ready for other people to use.
Yeah, good idea.
Might be ready.
And I have a request, and that's that I'm a very back-end oriented person, and so I'm always interested in workflows and timings of how you did something and how long it took and all that sort of stuff, and what was good and what was bad and all the sort of parts that people don't show.
Okay. Very good. Okay, well, hearing nothing further, I guess we will adjourn. Thank you all for joining, and look forward to next week then.
Thank you so much, David. Thank you all.
Thank you very much, Frank, for your presentation.
Thank you.
Bye-bye.
And thanks to Victor again for providing the Zoom.
Pleasure.
Thank you, Victor.
Bye-bye.
Thank you. Take care.
Goodbye.
Bye.
Bye.